following video is one of 5 recordings from the Environmental Lands Management Field Day presented by the University of Florida IFAS South Florida Beef Forage Program on Thursday, March 31st, 2022 at the UF IFAS Range Cattle Research and Education Center in Ona, Florida.
Good morning, everyone. Feel free to stand up. I know it's early. If you need to get coffee, if you need to use the restroom, and feel free also to interrupt me during my presentation. My name is Maria Silveira. I'm a faculty here, soil scientist. I've been with University of Florida for about 16 years. And today we're going to talk about nutrient management. I think there's a lot of misconception about nutrient management and how ranchers manage nutrients in their land. So that's the main goal of this presentation, to just share with you some of the information that producers are required if they're enrolling BMPs. So it's not, I think there's a general perception that they are simply overusing fertilizer, which in the reality is very rarely they can even afford fertilizer. So when they can afford fertilizer, there are certain tools that they use to determine what type of fertilizer, where and when we can apply to those pastures. So usually the questions that especially this year that the fertilizer prices are incredibly high, questions that I get more often are, what else can I do besides commercial fertilizer? Or can I go by a year without a fertilization? That's mostly the most common case in Florida. Even here at the research center, Dr. Sellers mentioned we have about almost 3,000 acres of land to manage. We don't fertilize every single, in fact, we never fertilize a lot of our pastures, grazed pastures. So we have to target maybe the species that have a higher nutrient requirement, maybe hay fields, plant material, but definitely the grazed pastures are not our top priority. And of course, our native rangelands, they never received any fertilizer. So let's talk a little bit. What are we trying to accomplish? It's not moving. When we design a nutrient management, why in agroecosystems we have to add nutrients compared to a native land that never requires? Why we have to add those external inputs? Usually it's because the nutrient cycles are not in balance when we have agroecosystems. And the reason they are not in balance is because our goals are different. So whether we're removing more nutrients with the harvested crop, and we have to remember in a grazing situation, calf, the animal that is leaving the land, it's also removing a lot of nutrients. So this is a source of nutrient it's not escaping, but it's leaving the system. So eventually we have to supply the nutrient back to the land. Or we may have what I'm calling dislight leakages, which are not desirable. So they're unintended losses of nutrients, and then we're trying to minimize that as much as we can. Or we have plants that demand higher, like I said, higher nutrient levels. So if you have a blue stem and you have a Tifton 85 Bermuda grass, they have completely different nutrient requirements. One produces much more, and I'm going to show you some data. One that usually produces more, which is desirable from a production perspective, they also require higher levels of nutrient inputs. We also may have different increased animal density. So again, animals are consuming the forage. We have to make sure that the nutrient requirement, the forage is met so the animals can have the adequate amount of forage. So, and we're talking about nutrient management is not just soil testing and tissue testing, but for the purpose of today's presentation, we're going to talk a lot about soil tissue testing. But I want to just mention, if a producer is enrolled in the FDAX BMP program, they're required to do a bunch of things that goes beyond just testing the soil and the tissue. They have to know what the soils are in their different pastures. Many times they are required to do a complete nutrient budget. And I have some issues with nutrient budget. This is not the purpose of this presentation. I'll be happy to share with you an article that I wrote, why nutrient budget doesn't work on a farm level. But anyway, they have to know what rates and what sources of fertilizer they're going to be using, the timing that those nutrients are going to be applied. But like I said, for the purpose of today's presentation, we're going to be focusing on those items: highlighted in yellow. So soil fertility, which is very interesting to me. I'm a soil scientist. I got my PhD in soil science, and then I moved to Florida. And I said, really? This is what we're dealing with? Soils in Florida? It's a completely different nature. That's why even for somebody that had a degree in soil science, we have to relearn certain things in Florida because our soils are what? Extremely sand, low organic matter level, retains very little nutrients. So we are either too wet or too dry, retains very little nutrients, water holding capacity, very little. So the way we manage soils is a little different. Usually when we talk about nutrient management, is not increasing nutrient levels in the soil. That becomes quite costly and very ineffective for producers. So the goal is not to bump up my phosphorus level from medium to high. Never, this is not the philosophy that we use in Florida. In other states where we have, I'm going to say real soils, I'm sorry, Florida soils, they can afford to do that. But in our situation, very rarely we can afford to do that. And our range, our flexibility is very limited because we can also mess up too much because we're already dealing with something that's very poor. So if we overgraze, like Krista said, any physical disturbance, we can lose that little bit of organic matter that provides so many benefits to the soil. Then we're in trouble as well. So plants require 17 essential nutrients. You may have seen this, or you may have encountered, and we have a lot of those snake oil products that they say, well, but my product has 65 nutrients. So what? Plants only require 17. Doesn't matter if you have 20, 30, 40, doesn't matter. A lot of these nutrients, especially in a perennial crop, everybody here had a chance to dig a hole in a pasture to see the root system? It's pretty extensive. I mean, some of the bahia grass, for instance, if you can go as deep as you can dug the hole, you're gonna find bahia grass roots. So a lot of these nutrients are present in the soil in quantities that doesn't require additional application. So for instance, if micronutrients is a good example. By the way, the micronutrients are the one that I believe it's highlighted in green. My screen looks different than yours. So I apologize if that's not the same color. I'm colorblind from here. So the chlorine, boron, iron, these are all micronutrients. We very rarely have to worry about micronutrients in a grazing situation. The most common ones, and I guess I can use this better, that we usually refer to are the nitrogen, phosphorus, and potassium. Those are primary macronutrients. So when you producer buy a mixed fertilizer formula, 10-5-5, that means it's 10% nitrogen, 5% of phosphorus, and 5% of potassium. So we're referring to nitrogen, phosphorus, and potassium. That doesn't mean they're more important than the rest. It's just that plants usually require larger quantities than the others. Why we don't worry about calcium, magnesium? Because usually when we lime the soil, apply Dolomite or any liming materials, those contain calcium, magnesium. So automatically we're supplying the plants with calcium, magnesium. Sulfur is also very rarely an issue in our soils. With that in mind, and let's focus on the macronutrients and NP and K. You see this table. I have some of the most common forage species that we use in Florida, and here the potential yields in tons per acre. You see yields are usually high for Bermuda grasses, but like I said, they also require higher levels of nutrients to maintain the higher production level. So if you look at this, is how much is accumulated in above ground tissue. So if you compare Bermuda grass, jigs, Bermuda grass, for instance, bahia grass is almost twice as much nitrogen as bahia grass because it produces twice as much biomass. The same is valid for phosphorus and potassium. So when producers call us and ask what is the best forage to grow, there's not and we say that our answer, of course, in an extension, our answer is always it depends. But in this case, it's really true. It depends. It depends on the level of input that the person is willing to put. So if they treat a Bermuda grass field, that can be jigs, can be Tifton 44, Tifton 85, any of the high yielding cultivars or species, if they treat the same way that they treat Bahia grass, they either, best case scenario, they will end up with Bahia grass, or worst case scenario, they may end up with something else, any type of weed. Then they will call Dr. Sellers to address those weed issues. So the level of input will determine what is the best grass for a particular operation. Of course, there's other factors, how wet the soil is, but from the nutrient management perspective, that's usually the main factor dictating what the best grass is. And of course, legumes, they can capture nitrogen from the atmosphere, so we don't have to fertilize legumes with nitrogen. But it sounds very easy. However, the reality is very difficult to maintain a stand of legumes, especially if it's intercropped with the grass pasture. It's a very challenging thing to accomplish. So this is our guide. University of Florida, we don't have any regulatory power. We don't tell producers what they have to do. We have this document that makes suggestions relative to fertilization, and is based on our research. But we understand that even though we don't have regulatory power, regulatory agencies use this information to implement some of the regulations. So if you read for instance, the cow-calf BMP manual, you're going to see that they refer to this document. This is called SL 129, or Standardized Fertilizer Recommendations for Agronomic Crops. So this document contains all the different fertilizer recommendations for different crops, different forage crops, and depending on how they are managed. So even for Bahia grass, that takes a lot of abuse in terms of not fertilizing, overgrazing. Even for Bahia grass, fertilizer recommendations will vary depending on how it's managed. Do they cut Bahia grass pastures for hay once a year? Fertilization management is going to be slightly different. Or it's grazed year round. Is it producing seed? Are you establishing a new pasture? So those are factors that will determine how much fertilizer and what type of fertilizer will be recommended. So again, going back to my initial comment that there's a lot of misperception, I think the general public think that producers every year they buy truckloads of fertilizer, then dump to their pasture. And maybe there's some exceptions that they do. I never came across anybody like that, but we have tools that they can use to determine only, and these tools are based on a minimum amount of nutrients that will be necessary to maintain a health standard. So we're not looking at maximizing production. No, it's what is the minimum that we can get by. And that's exactly what the document describes. So even what I mentioned, this is the minimum. A lot of times producers cannot afford. So if you look at, for instance, Bahia grass, we recommend anything between 50 and 160 pounds of nitrogen per acre per year. I've never seen, 16 years here, I've never seen anybody that applies more than 80 pounds of nitrogen. So the recommendations are there, but the reality is usually producers are in the lower end of those recommendations, or if they can afford. So it's just another example to show you that we have the tools that producers can use to determine how much and what nutrients need to be applied. Nutrients is important, but more importantly is the soil pH. Soil pH is a very simple procedure to be determined in the lab. I believe some county extension offices offer that analysis. Well, at least in the past they used to. But we're going to talk about labs. I highly recommend to use the lab because they provide the complete analysis. But anyway, it's something relatively simple and is the basis. So for instance, this year, we cannot afford fertilization, nor even in our hayfields here at the center. How we prioritize. pH is the first factor determining where we're going to apply fertilizer or how we're going to use our resources. We much rather use our resources buying lime and increasing the pH of a pasture that's below that recommended rate versus applying nitrogen to a pasture. So pH is the basis. Once the pH is corrected, then we start thinking about the fertilization program. The issue with pH is most of the times the native soils are highly acidic, talking about our nice soils in Florida. Usually native soils in Florida are very acidic, which means that we have to lime to bring the pH to those desirable ranges. The good thing is it doesn't take much lime to bring the pH up. But sometimes those lime materials take a little while in the soil to react. So if somebody calls me now, we're in April, that's when most of the producers will be fertilizing. If they haven't done yet, they'll be fertilizing in the spring. That's when they need the critical period that they need forage. Let's say they test the soil now, which is too late. But let's say they test the soil and the test came back with a pH, let's say 4.5, very common. And they say, well, I want to fertilize. I need the grass I need to go grow grass this year. It's probably not going to be the best scenario, because the first thing you have to do is, again, going back to the basis, pH. So you have to raise the pH from 4.5 to 5.5. 1 unit means 10 times the acidity, because the pH is on a logarithmic scale. So that, and I mentioned that dolomite takes a while to react in the soil. So what I'm trying to say with this message is it takes a little bit of planning. Right now, if they realize, wow, I haven't done all of this, it might be a little, I won't say too late. It's better to do now than not do at all. But it will be maybe a wake-up call for next year when they have a better planning for next year. Because again, you need at least 3 months for liming material to react in the soil, bring the pH up. And if you apply nitrogen right after a lime application, you may lose a lot of the nitrogen by volatilization. They're not compatible soil amendments. Anyway, but how do I determine when and what type of amendments, including lime or fertilizer, are needed? And the answer for that is soil and tissue testing. It can be a very time-consuming process depending on the size of the operation. So if you have if you're managing a small operation, it's not such a big deal. But if you have 10,000 acres and I tell you have to test soil test, and I will provide you with information how many subsamples you have to take, that's a pretty major investment in terms of labor. But the benefit you get from that is, I think it's still worth. So why we test the soil? Even though you may have an idea what you are dealing with, the best way to accurately determine what nutrients need to be applied is by testing the soil. So, and I'm going to talk about some of the details about testing, but we also want to, if you do that, implement a soil testing program, and you have the results and you compare the results over time, you can see how your soil, your nutrient management is affecting the soil property. So maybe you see that, well, I limed the soil 2 years ago. Now I see that the result is still lasting. So it saves you in terms of the inputs that will be needed in the future. So when we test the soil, we're not determining the total amount of nutrients. We're determining what the plant sees. So the lab uses a procedure that mimics what the plant roots see as plant available. Again, going back to that idea of our very rich Florida soils, our soils, when people say, well, my nutrient, especially phosphorus, I hear that a lot, my phosphorus is tied up in the soil. So, and there's companies that sell products to make the phosphorus, release the phosphorus. That's not necessarily true for our soils. Again, our soils are very sandy, very low nutrient holding capacity. In fact, I wish that our phosphorus was tied up in the soil, because then the plants could better utilize. But usually our problem is the opposite. But now if you move to Georgia, where they have very clay soil, fine texture, yes, the phosphorus is tied up in the soil. But again, when we use, especially people that are trying to sell products across the United States, they use the same concept in Florida. I have a hard time. I wish that we had that problem in Florida, but our problem is actually the opposite. We want to retain the nutrients so the plants can better utilize before it runs off. So it's not a direct measure of total, it's just what is plant available at that time of the year. So and I believe that we're going to have a demonstration this afternoon about how to properly collect a soil sample, but very general. It's a simple procedure. We need multiple samples to represent the area that's being evaluated. We need same consistent depth. Usually it's between 4 and 6 inches. And we need consistency, if you can, in the same, collect the sample same time of the year, every year. So that's basically what it is. But you see that number here, 15 to 20 individual cores. So again, if we're talking about a large operation, that number can become pretty overwhelming. Unfortunately, we don't have the remote sensing tools. So there's tools now that people are trying to develop, companies are trying to develop, can hook up in the back of your tractor, and you can just drive in the field and you can map. This is being done for carbon, by the way. Krista mentioned carbon sequestration. Because of the carbon market, people are trying to do that. But the technology is still not there for nutrients. So we have to physically collect the sample, submit the sample to the lab. We are not there yet. Maybe 10 years from now, that this whole presentation will be outdated. But right now, unfortunately, we have to dig holes and submit the sample to the lab. And I highly, especially because it's a labor-intensive process, don't discard the samples until you get your results back from the lab. So keep a subsample in case you get funky results from the lab, which happens more often than we wish, but that way you don't have to go back and repeat the whole process again. How often we have to do that? It depends. If it's intensive productions and hay system, maybe every year. BMP manual will also dictate how often producers are required. Again, IFAS, we're not requiring anything, we're making some suggestions. In a grazed pasture, usually recommend every 3 years, the soil need to be tested. And I mentioned this the same time of the year because soil moisture levels can change some of the soil properties. So if we consistently collect a sample every fall, results are be more comparable over time. And I mentioned the issue with the pH, then if it needs to be corrected, it takes a while. So basically, when we submit the sample to the lab and the lab sends us a result back, it's not the absolute number that we're interested, but it's more the range that we're talking about. And this is how, I don't know how many of you seen soil test report. I'm gonna show you one at the end of the presentation, but your nutrients will be rated. In this case, right now IFAS use these 3 levels: low, medium, and high category. Usually whether it's phosphorus or potassium, when it is medium category, that means there will be no additional crop response if fertilizers is used. So that means that the yields, it's my mouse. Okay, I guess it doesn't show. The yields will be in the yellow range, that you don't get an increment in yield if you apply the fertilizer. So the same concept is valid for tissue testing. But the issue we have is, you see those numbers on the Y. I'm gonna step out here. You see those numbers here, 530, 60? It's a specific test that we use to determine what those numbers are. If a producer sends a sample to another lab that doesn't run the same procedure, the same soil test procedure, we may get numbers that are different. Or the lab may rank, the procedure might be the same, but they may assume that 30 instead of being low, or I mean 60 instead of being median, they may assume the lab may interpret that as being low. So that will trigger a fertilizer recommendation. So those are the things that we need to be careful when if a producer ever asks you help with soil test interpretation, these are some of the things that we need to look for. So for instance, for phosphorus, these are the ranges and it usually is a range, it's not a number. So it's not a clear breakdown. Usually it's a range. Like I said, for medium phosphorus, for instance, anything that has between 26 and 40 parts per million. That means that if the soil test is above that 26, no phosphorus recommendation will be, no phosphorus fertilizer will be recommended. There's a little caveat for barrier grass, and we're gonna talk about, because it also requires tissue testing. Like I said, now labs offer a wide range of soil testing procedures. You may test labs offering soil health assessments. And we can talk about soil health, but we're talking about strictly in this presentation about fertility testing, which is fine. Just again, we need to keep in mind that the tests need to be compatible with University of Florida recommendations. Otherwise, we have trouble comparing those results. And then units can be reported in different ways. The index, as I mentioned, those numbers, lab may interpret it different ways. So what I did a few years ago, I collected one sample, homogenized that sample, and I sent to 5 different labs, commercial labs, the same sample. And here are the methods that each of the lab methods that the labs ran. By the way, our standard soil testing procedure in Florida is called MELIC 3. So the number 1 and number 2 and number 4. And then and the rest of the table is showing the report that came from these 5 different labs. I mentioned the pH is the basis and a very easy procedure. You mix soil with water, mix for 5 minutes, and you insert a probe, and that measures the pH. So even for an extremely simple procedure as pH, you see the range of pH values that we got from that same sample. What that means is, for instance, lab number 3 will trigger liming recommendation. But now if I send the same sample to laboratory number 5, that won't be a liming recommendation. So which one is the correct one? More importantly, I'm gonna move to the next slide. So not only the numbers and the rating were different, but also the fertilizer recommendations. Usually commercial labs, they tend to have higher fertilizer recommendations. Producers question us all the time. Why, if I send my sample to Georgia, for instance, there's a laboratory in Georgia that's very popular, which is fine. They do a very good job in terms of analyzing. But when you look at the fertilizer recommendations are much higher than University of Florida. The reason for that, and I'm gonna move to the next slide, but you see the differences. It can be number 2, by the way, is University of Florida lab. So recommends 50 pounds of nitrogen to the acre because the producer, when they submit the sample, they have to indicate what crop it is. It's Bahia grass, I'm grazing, I'm in the low input end. And you see there's no phosphorus recommendation because I mentioned that there's a caveat for Bahia grass. It requires tissue testing. And then if you went to with the lab number 1, there's a 40 pounds of P2O5 that may not necessarily be required, but it will be an additional investment that producer might make that will not translate into greater forage yield. So these are some of the things that to keep in mind. I'm not saying that we have to submit samples to University of Florida. We just need to be careful with the fertilizer interpretation. And I mentioned why those commercial labs usually suggest higher fertilization rates is because of what I mentioned in the beginning is the fertilizer philosophies. Different fertilizer philosophies are used across the United States. Soils that can retain nutrients a lot of times, especially for annual crops, that they need immediate supply of nutrients during the critical periods where corn is flowering. So it's not the same as a perennial crop like Bahia grass. So the philosophy is to increase nutrients in the soil so the soil can supply the nutrients when the plant needs. Again, going back to our rich Florida soils, that's not the case, especially for a crop that grows year long, like Bahia grass or forage grass crops. So our philosophy is just to supply is this first one, sufficient level of available nutrients. So basically you are supplying the minimum amount of nutrients that those crops can require so they can complete that. And it's just for one growing season. So this is basically why the difference. In the past, I don't think these labs are using, they also use this cation saturation ratio. So there's this perception that if you have cations are the positively charged nutrients, potassium, calcium, magnesium. They are in a certain ratio in the soil that will be the best environment for the plants to thrive. There was very little science behind that concept. I don't think labs use that. Once in a while, I'll come across somewhere where they say, well, you have to have this specific ratio, calcium, magnesium, potassium. There's, like I said, very little science behind that. So we don't use that in Florida. And I mentioned that I asked, have you ever seen a soil profile and had you had a chance to look at the root depth? And that's exactly the limitation. So I said, well, we need to do, but here's the limitation. So when we sample the soil, I mentioned that we only collect the 4 to 6 inches. So that top layer on your right-hand side in the picture, you see there's a lot of roots in that surface, but I'm gonna step out. And you see the dark color. That dark color, and this is most typical soil, Central South Florida, is photosol. The way this soil was formed in our landscape, a lot of nutrients leach from the surface layers into this sporic layer, the dark layer. So you have a gradient in nutrient concentration, especially phosphorus. So you may test the soil on the surface. You might get a 5 parts per million extractable P. As you go down the soil profile, and if you test that dark layer, you may get something like 50, so 10 times more. Plants can utilize that phosphorus, you can clearly see the roots, but how can I effectively determine how much that entire profile, how much the plant C is available? There's no way we can tell producers, well, all you have to do is dig 15 to 20, 40 inches hole in your pasture and submit. No, they'll look at me and say, she's kidding, right? So what we came up as a compromise, we came up with this tissue testing. So the tissue is like a blood test. It pretty much tells what's going on with your body, the same with the plant at that specific time. So we developed this critical tissue level, again, looking at the minimum. What is the minimum phosphorus level in the tissue that will determine that the plants are deficient in P? That's why we developed that tissue. So for Bahia grass, we use tissue, soil testing, and tissue testing to determine when phosphorus is needed. It's been a very effective tool. Of course, it's another tool that every time that we suggest something new and then there's labor involved, we encounter some resistance. But now it's being incorporated in the BMP, and this is how we've been managing phosphorus fertilization for established Bahia grass pasture. So I just want to show, just explain the rationale behind. And let's see the last. So this is basically the interpretation for, and again, this is only valid for established Bahia grass, but because Bahia grass is such a common species in Florida, most of the situations we're going to run into this. So if a soil sample only submitted to the lab, and if it tests low in P, what the soil test report is going to tell is you need a tissue test to generate a phosphorus recommendation. If the soil is median or higher, then there's no need for testing the tissue. We know that the levels in the soil are already sufficient to meet the Bahia grass requirement. So the critical tissue level is 0.15%. So you can have a soil, let me explain what is the most logical. You can have a soil that tests low in P. Let's say 99% of the soils in Florida are going to be testing low in P. And then you may have two different scenarios. Tissue P is above that 0.15. That means no fertilizers required, because most likely the plants are getting phosphorus from deeper soil layers. That's not only that 4 to 6 inches that we sample. Or you may have a scenario 2 where the tissue P levels are below that critical limit, that critical 0.15%. That means that in that situation, that definitely phosphorus will be recommended. Whether person can afford is a different story, but that will be the recommendation. How much P will be recommended? It depends on how much nitrogen. So nitrogen is the major driver of production. More nitrogen you add, Bahia grass production increases almost linearly. So if you're using very low nitrogen levels, you're not pushing the plant, phosphorus recommendation will be low. It will be in that 25 parts P2O5 per acre. If you're using higher nitrogen levels, then the plant will also require higher P levels. That's basically the reason why we have those different numbers. So I want to go over this soil test report because there's a lot of confusion. So I blacked out the producer information. This is, if they submitted, this is not a recent report, but I suspect it hasn't changed that much. So the first, well, let me step back a little bit. When the sample was submitted to the lab, whoever submitted had to indicate which crop it is. So in this case, crop Bahia grass. So they indicated that it was probably established Bahia grass pasture. So that's how the lab will determine what is the target pH. If it was Bermuda grass, which is a different category, target pH will still be the same. And then you have this pH, which is, I'm sorry, I'm using my mouse and I forgot that you cannot see. So the target, can you all see the target pH here? Target pH is 5.5 for the particular crop. If the producer indicated it was a perennial peanut, the target pH will be different. This is the actual pH of the sample, 4.5. So the 4.5, how do I, it's below the target. But how much lime, how do I determine how much lime needs to be applied to raise that pH from 4.9 to 5.5? A lot of people say, well, 1 ton of Dolomite to increase 1 pH unit. Maybe 30% of the time that rule works, but I mean, it's a very wild guess. The most accurate way to determine how much lime is required to bring that pH from 4.5 to 5.5 is based on this other number, this buffer value, which is this number. A lot of people don't understand what that buffer means. That means, well, what it can buffer is the ability, is a concept, ability of the soil to resist to change. So changing pH in this case. Soils that contain more organic matter, they resist more to changing that pH. Soils with more fine texture, more clay, they resist more. Going back to our very rich Florida soils, low organic matter, low coarse texture, they have very low buffering capacity. How do we measure that buffering, that resistance? We use a solution. So this is called Adam-Evans. This is the AE. Maybe I'm giving you too much information, but I would be curious if I say, what is it? Damn AE in my report. It's an abbreviation for Adam-Evans. And this is the solution that has a pH of 8. So they react the soil with the solution and they measure the pH after 5 minutes. Depending on how much that pH dropped, it determines, it's indicative of the soil buffering capacity. So if you, in this case is 7.2. If you had a soil, the same soil, let's say same pH, 4.5, but the buffer was the buffer was, let's say 7. That means that the soil dropped the pH more. That means that it will require higher liming application rates. So this is the number that will determine how much lime will be required to reach that target pH. A lot of commercial labs, unfortunately, don't run that test. But this is how a lot of land-grant universities lab make that determination. Okay, I said pH is the most important thing. How about the nutrients? Again, melic, 3 extractable, and the results here are in parts per million or also milligrams per kilogram. Be careful that if you send to a commercial lab, you might get the reports in pounds per acre. We can, if it's melic 3, we can convert from parts per million to pounds per acre. But in this case, the soil test high in P. So when you look at the bottom, the fertilizer recommendations, won't be any tissue testing. Or in this case, I think the producer should meet a tissue testing. You see the result here. The soil that has high P, there is no tissue testing because the soil only provides all the results that we believe that. But in this particular case, the producer should get because it is what the soil is going to test. And the tissue P was 0.27. Remember, the critical is 0.15. So that means that it just confirms that the soil is supplying the plants with sufficient amounts of phosphorus. Then we look at potassium, very low, low. And then again, depending on how much nitrogen producers using, that will determine how much potassium will be recommended. Again, calcium, magnesium, all the other nutrients usually not included in the recommendations because if they're liming the pasture, it's also providing calcium and magnesium. Here's another example. I think it's essentially the same. Only difference is the target pH here was pretty close to the actual pH of the sample was pretty close to the target pH. So not always the lab will generate a lime recommendation. I think for University of Florida, every time, if the target is 5.3, for instance, every time the soil tests below 5.5, every time the soil tests below 5.3, this is a computer-generated report. The computer will generate a lime recommendation. Of course, there's somebody that oversees the computer generated and signed, so they reviewed the report, make sure the computer didn't mess up anything. But very similar. The only reason why I want to show this is to show that even the pH is below the target is not sufficiently low to trigger a lime recommendation. And that's where maybe the university extension faculty can help you determine, well, is it really needed? Can I go by 1 year? We know that if we don't do anything next year, probably the pH is going to be slightly lower. Just naturally, pH, rainfall, and other factors we have in Florida, tendencies for the pH to lower over time. So maybe it will be recommended, my recommendation in this particular case would be to test the soil the following year, or at least 2 years, make sure that the producer is monitoring that change in pH. I think this is my last slide. Oh, yes. I want to show, I found this very interesting. So I get a lot of calls about, especially when we start having algae blooms and people were concerned about fertile agriculture runoff, nutrients that escape. Again, that perception that we're overutilizing nutrients, which is this extremely expensive resource that nobody can even afford. But anyway, there's this perception that we have a lot of nutrients in grazing lands, and this nutrient somehow are escaping the system, is ending up in our waterways, and is causing all these water qualities, algae bloom and everything else. So the cattlemen, a few years ago, they asked me to calculate a budget, of nutrient budget, specifically for phosphorus. They want to know if the cattle industry was a net importer in the state level. So the whole state of Florida, is cattle industry a net importer or net exporter of phosphorus? And we're still, I don't have the answer yet. We're still working with the data. It looks like, well, I'm not going to say anything until I have the data. But one of the things that we often miss in budgeting, especially when we look at the whole state, is the human component. As human, we have a lot of nutrient imports into our watersheds. So the reason for this map is our population in Florida is about the same size as population in Australia. I'll use Australia because I lived in Australia for a little while. And this is the image that I'm showing is proportion, the area of Florida relative to Australia. So we're squeezing almost the entire population of Australia in this narrow strip of land with these very rich soils that I've been mentioning about that have wonderful ability to retain nutrients. So no wondering why we have so many issues in Florida. If Florida was a country alone, and the population, if you look at the population, it would be the 90th most populous country in the world. So we have an incredible amount of people living here. We all enjoy the landscape. No wondering why people want to move to Florida. But what I want to say is that we are part of the problem. And of course, we have to be part of the solution as well. So every time that we buy pet food, or we buy food for ourselves, or we buy even non-food, our cleaning products, we are importing a lot of phosphorus, and that phosphorus doesn't disappear. It ends somewhere. And I think with this exercise, what I'm going to show is, once we complete this project, is that there's a lot of unaccounted phosphorus that's entering the state through this high population. So when we blame certain commodities or certain sectors, is the water quality caused by agriculture, urban, or I think we need to look at a bigger picture, how we can solve the problem, not trying to blame individuals for that problem. But anyway, that's a just short story about this project that we've been working on, and how important it is to account all sources of nutrients, not just agriculture, but everything that we use. I think this is my last slide. Thank you so much. I'll be happy to take any questions if we have time, or in the afternoon, I'll be the whole day. We can also visit in the field. Yes, sir. I'm going to repeat the question because folks that are watching later may not. So the question was how to handle pH when it's high. And that's a very difficult situation. First, we need to figure out why the pH is that high. Maybe something was overused. A very common cause of high pH is excessive biosolids, lime stabilized biosolids. In the past, a lot of areas received biosolids based on the nitrogen content. And I don't think that folks fully appreciate the fact that the pH was increasing over time to a level now that I visited some places in Central Florida, it looks like a desert, not even weeds grow. So how we handle that? We have this lovely question. A lot of the county agents here had to deal with this situation. And in 1 year, I can guarantee you're not going to be able to do anything. 1 year is a very short, fixing that problem is a long-term process. So a couple of things that we recommend. There's not unfortunately, I wish I could say, well, do this and problem solved. Depending on what cause, it could be multiple years of excessive, like I said, lime stabilized biosolids. I'm not saying that biosolids by no means is a bad source of nutrients. It is an excellent source, but when it's overutilized, anything in excess can be an issue. So a couple of things that we recommend: testing the soil at deeper depths. So is the deeper soil, is the pH consistently high throughout the entire soil profile? Let's say, oh, it's only high in the top 4 inches. Once you go down, it's not that high. So mixing the soil, very expensive. You have to reestablish a pasture. I understand that. Changing the species. If it's not excessively high, by excessively high, I mean you're not in the 8s. If you are in a more manageable 6.5, 7, change the species. Bermuda grass is less susceptible to high pH versus Bahia grass. So maybe considering a different forage species. Finally, and this is again, it's not going to change over time. It's not going to solve the problem over time. It's using fertilizer sources that have a greater acidifying potential. By that, I mean ammonium sulfate. Ammonium sulfate can acidify the soil, but it's not going to change the soil in a year. May not change in 2 years, may not change in 3 years. Might take, there's producers here in Polk County, they've been dealing with that for 10 years, right, Bridget? And they still see very little change in the pH. So unfortunately, that's not the answer that you probably want to hear, but it's very specific to the site, and the reason why that pH increase will dictate how long it will take to fix that problem. What will be the other cause of, I don't know. What the other cause of a high pH other than lime stabilized? I don't know. Water, irrigation water, usually in citrus can be an issue if the pasture used to be an old citrus grove and it was converted to a pasture. Irrigation water, they pump water from the groundwater, the pH can be high. That is a little easier to fix if it's water related. But other than irrigation water and then lime stabilized, I don't know any other cause. You're welcome. Any other questions? Well, if not, I'll be here the rest of the day. I'll be happy to visit with you. Thank you so much.